My channel Sequential Biology. In this video we will discuss about phylum Porifera and their classification up to subclass. First we will see general body plan of a common representative of this phylum, that is the sponge. And the organisms or the species of this phylum, usually they reside in marine water, but some freshwater form can be also found. And they are the diploblastic organism, and since they have no tissue grade of organization, different type of cells are present in their body, and this different type of cell perform different type of function. And you should know about their position. Then this is the outer layer of this phylum, and which is known as epidermis. The outer layer consists of tightly packed epidermal cell. Then this outer layer is made up of many epidermal cells which are tightly packed. And in this epidermal layer there are present many small pore, and which are known as ostia. Then these are the ostia, and sea waters enters through this ostia, and ultimately the sea water comes in a central cavity, and which is known as spongoshell. And this is a characteristic features of this phylum. The presence of spongoshell is a characteristic features of this phylum. Then the water passing through the pores enters a cavity which is known as spongoshell. Inner wall contains a special type of shell, and which is known as choanocyte, and this is a choanocyte cell, and this choanocyte cell have a characteristic features. These choanocyte cell are also called as collar cell because they have a collar like structure. This is a collar like structure, and for this reason this is also known as collar shell. And this collar cell is present in inner wall of the animals. And I have already told you that they have two layers of cell, the outer wall and the inner wall. Then the position of the spongoshell is inner wall. They reside in the inner wall just adjacent to the spongoshell. And also in this collar cell there are present a large flagella. So this is a flagella, and the beating of this flagella creates a water current, and this water, the water which enters to the ostia, enters into the spongoshell, and this beating of the flagella creates a water current inside the spongoshell. And this will causes the enters of the food particle into the collars, and then ultimately the food particles enters into the choanocyte cell. Either these food particles will be digested in this choanocyte cell, or this may be transferred into the amoebocyte cell. Amoebocyte cell is present in another layer which is present in between the two main layers, the outer epidermis and the inner endodermis, and in between these two layers there are present a mesohyl. So mesohyl is a gelatinous structure. There are present different type of cell, and these cells are originated from the amoebocyte. The amoebocyte, these cells can transport the nutrients to other cells. There are present any other cells apart from this amoebocyte. Then the amoebocyte, after getting food particle from the choanocyte cell, may also transfer these food particles to another type of cells which are present also in the mesohyl layer. And also another important function of this amoebocyte cell is to produce a materials of the skeletal fiber which is known as spicules. This is also an important characteristic features of this phylum. So you can see there are no tissue grade of organization. Different type of cell perform different type of function. We can say that the sponge are most primitive animals, and they cannot move from one place to another. They are sedentary. And since the presence of these choanocyte cells, and these choanocyte cells shows a resemblance with the choanoflagellate, and for this reason it is believed that the sponges are developed from a choanoflagellate like ancestor. This is basic idea of this phylum. Now we will discuss about characteristic features of this phylum. Phylum Porifera. The term porifera comes from two Latin words. The porus means pore, and fera means beard. That means the animals whose body contains many pores, they are called as the pore bearer, or they are included under phylum Porifera. Presence of choanocyte cell or the collar cell reveal that the phylum Porifera evolved from choanoflagellate like ancestors, because this choanoflagellate actually looks like collar cell, and for this reason it is believed that this phylum Porifera originated from choanoflagellate like ancestor. This type of collar cell can also be seen in case of Cnidaria and few Platyhelminthes. Then this phylum, or the representative of this phylum or the species of this phylum, are most primitive multicellular organism, and they do not move. And for this reason Aristotle, Pliny and other ancestor naturalists believe that they are plants, because plant cannot move from one place to another, and also the animals do not show any definite form And for this reason, the ancient scientists believed that they were plants, and sponges are filter feeder, rely on water current for food, and the filter out food particle from the surrounding water. Usually marine and found all over world. Then the species of this phylum are present all over world, and they are usually marine, but and some forms may also are freshwater form, and they found in all depth of ocean but prefer shallow water. Shallow water means where the water level is not deep. This is called as shallow water. The animals of this phylum present in all the depth of ocean, but usually they prefer shallow water. These are the basic characteristic features or the general idea about this phylum, and now we will see the phylum character. And this phylum shows asexual reproduction by bud and the gamules. This is very important in examination point of view. The asexual reproduction by budding and formation of gamule. Now, generally they are the marine and the asymmetric. Asymmetric means they have no definite shape, and also sessile, that means cannot move from one place to another, and occurring singly or in colony. Singly means single individual can be found, or also colony means many individual found in a specific region. This is called as colony, and the few are also freshwater. The spongocele, numerous ostia or pores present throughout the body, and present many different pores, small pores, and these pores are known as ostia. So numerous ostia or the pores are present throughout body, and a large opening. This is a large opening which is called as osculum, found on upper end. This is upper end. So in upper end you can found a large opening, and which is known as osculum. Sponges have a water transport or the canal system. This is characteristic features of sponges, phylum porifera. And in case of echinodermata, we can see water vascular system, but here in case of this phylum we can see canal system or a water canal system. This is the basic differences in between echinoderm and phylum porifera. In phylum porifera we can see canal system, but in echinodermata we can see water vascular system, and that means the water vascular system not only helps in feeding but also help in their circulation. And this water vascular system is seen in phylum echinodermata, right? Water enters through minute pores or the ostia in the body wall into a central cavity which is known as spongocele, from where it goes out through osculum. And these are the different layers in their body, and this is outer layer, the outer layer, and in this outer layer many minute pores are present which is known as ostia. We have already seen the position of this ostia, and just after ostia there are present a gelatinous layer which is known as mesohyl. And since there are present only two layers, the outer layer and this is inner layer, and for this reason they are actually diploblastic. But in between these two layers, a gelatinous matrix is present. This is known as mesohyl, and different type of cell are present in this mesohyl. And also you can see their skeletal system, that is the spicules, and these spicules are formed from amoebocyte cell, and these spicules are also present in this mesohyl layer. And the collar cell are present in inner wall, just lining of this spongocele. This collar cell line the spongocele layer, and the flagella of this collar cell creates a water current in the spongocele. Transport canal system are present here, and the waters enter through minute pore or the ostia in the body wall into a central cavity is called as spongocele. They have a central cavity. This is known as spongocele, from where water goes out through osculum. This is osculum, and the water enters through ostia and then ultimately come out through osculum. Osculum are present in upper part of their body. Sponges produce antibiotic. This is extra information. And the cribrostatin. What is cribrostatin? A compound isolated from marine sponges kill the cancer cell and the penicillin resistant streptococcus bacteria. Then cribrostatin is a compound or the chemical, and this is isolated from marine sponges and kill cancer causing cell and penicillin resistant streptococcus bacteria. Next level of organization, they are multicellular, because we have already seen that different type of cells are present in their mesohyl layer, and also collar cell are present in their inner wall. So they are multicellular but with few cell types, but the type of cell are few. Not a huge number of cell or huge type of different type of cells are not present there. Only special type of cells are present in this phylum. The body of sponges consisting of two layers of cells separated by gelatinous layer which is known as mesohyl or the mesenchymal layer. The outer layer is called the dermal layer and the inner layer is called as choanocytic layer. This is called as choanocytic layer because the choanocyte cell are present only in inner layer. This is very, very important, the position of this choanocyte cell. Since this shows the cellular level of organization, so there are present different type of cells, and the most important type of cells are choanocyte is lined with the choanocyte cell, and this choanocyte cell are flagellated, and the flagella covered by finger-like projection like the collar, so called the collar cell. These choanocyte cell are also called as collar cell because we have already seen that if this is a collar and the flagella are present, so this is the general structure of this choanocytic cell. Beating of flagella filter the food particle from sheep water, and these food particles enters into this cell and enters into this cell, that means the choanocytic cell, and then this choanocytic cell convey this food particle into adjacent amoebocytic cell. This creates a water current that draws water through ostia and out through osculum. The choanocytic cell is a leading cell controlling the water canal system in sponge. Amoebocyte cell. Amoebocyte cell are also present in mesohyl layer. This is not present in inner layer. This is present in mesohyl layer. And they use pseudopodia, and for this reason they are known as amoebocytic cell. They can move through mesohyl. Since they are present in mesohyl, so they can move from one place to another with the help of their pseudopodia and take up food from surrounding water, and they take food from surrounding water and also from collar cell and digest it and carry the nutrients to other cell. So this choanocytic amoebocytic cell provide nutrition to another type of cell, and they produce the skeletal fibers or the spicule. This is an important function of this amoebocyte cell. This amoebocyte cell produce skeletal fibers or spicule, and they are totipotent because other cell types. Totipotent means a cell when differentiated into another type of cell, then this cell is called as totipotent cell. Then the amoebocytic cell is totipotent cell found in phylum Porifera. And skeletal system. The skeletal system present in mesenchyme or the mesohyl, and the amoebocyte cell produce the tough skeleton fiber in the mesohyl. Then their skeletal system is made up of tough skeletal fibers. And what are the component of these fibers? The fibers may be made up of calcium carbonate or the calcareous, or maybe silica or the siliceous. Then the fibers which are synthesized from this amoebocytic cell are two type, made up of calcium carbonate, and when this fiber is made up of calcium carbonate, then this is called as calcareous spicule, and when this is made up of silica, or then this fiber is called as siliceous spicule. And many sponge produce more flexible fibers composed of the protein called the spongin. Many sponge produce more flexible fiber. So this, and apart from the calcareous and the siliceous spicule, most sponge produce more flexible fibers. This is more flexible fiber. Calcareous and the siliceous spicule are rigid fiber, but the flexible fiber, many sponge produce a flexible fiber which is called as spongin. Actually, this flexible fiber is made up of a protein spongin. This is also called as spongin. Then their skeletal system. Are two types. One is tough skeletal system, and this tough skeletal system is made up of calcium carbonate or silica, and also there are present the flexible fiber, and this flexible fiber usually made up of protein, and this is organic molecule protein, but this calcium carbonate and the silica are inorganic molecule. This is an important point. You should understand the differences in between this flexible spongin fiber and the calcareous and the siliceous fiber, and their digestion is intracellular. And we have already seen that the collar cell digests the food particle, and also amoebocyte cell digest food particle. So their digestion is intracellular. They have no respiratory and excretory organ. Primitive nervous system made up of network of neuron. Nervous system can be found in this phylum, but this is primitive and only made up of network of neuron. This different type of neuron form a network like structure. Then this is a very, very primitive nervous system which first evolved in this phylum. They are hermaphrodite. Each individual function both as male and female. A hermaphrodite, that means they are monoecious. Each individual function as both male and female. Male and female are not separate. Usually, a single animal serve both as male and female, but the cross fertilization can be seen. And just like in case of Ferritima, and the Ferritima are hermaphrodite, but cross fertilization seen in this Ferritima, because for fertilization two different individuals are needed, although they are hermaphrodite. And asexual reproduction by birds and gamules, this is also an important characteristic features of this phylum. Now the larval form, amphiblastula larva found in case of Cycon and the parenchyma larva found in Leucosolenia. The two important larval form are amphiblastula larva and the parenchyma larva. Then the amphiblastula and the parenchyma larva are characteristic features of this phylum. We will see the classificatory scheme of this phylum. Then the phylum porifera divisible into three classes: the Calcarea, Hexacti, Annelida, and Demospongia. The Calcarea again divided into two subclasses, the Calcaronia and the Calcinia, and the Hexacti Annelida divisible into two subclasses, the Hexosterophora and Amphidiscophora, and the Demospongia divisible into two subclasses, Tetractoniomorpha and the Seractinomorpha. But we will focus only up to this class, okay? And in different type of exams, you have asked the questions up to class level. We will discuss the class characteristics of this phylum. The first class is Calcarea. This is a representative of this class. The Cycon. Cycon is an example of class Calcarea. They are exclusively marine. Not found in freshwater. And the skeletal made up of Calcarea spicule. Their skeleton made up of Calcarea spicule. That means in their mesohyl layer, the Calcarea spicule are present, and this Calcarea spicule are hard skeleton, and this is developed from amoebocyte cell. Amoebocyte cell secrete this Calcarea spicule, and this Calcarea spicule are present in their mesohyl layer and show their body is very much rigid. And this Calcarea spicule is made up of calcium carbonate, calcite or argonite. Choanocytes are relatively large. Their choanocyte cell are large size, and the osculum narrow and provided with osculum fringe. The example of this class is Cycon or the Scypha and Leucosolenia. Second class is Hexacti Annelida. Hexacti Annelida, the skeleton consists of 6 red siliceous spicule. That means the siliceous spicule means their spicule is made up of silica, but in case of class Calcarea, we have seen that their skeletal system is made up of calcium carbonate, right? And for this reason, they are called as Calcarea, because their skeletal system is made up of calcium carbonate. But in Hexacti Annelida, their skeleton is made up of 6 red siliceous spicule. 1, 2, 3, 4, 5, 6. 6 red siliceous spicule can be found in this class. And the choanocyte is small. Choanocyte small and entirely marine. They are also entirely marine, and many found in the deep sea. Body shape is carp urn on the face like, and the spongocoel open by a wide osculum. Their osculum is wide. And the example of this class is Euplectella. Euplectella is also called as Venus flower basket and the Hyalonema. This is very important example. Glass rope sponge. The glass rope sponge or the Hyalonema is an example of class Hexacti Annelida. And here you can see the diagram of Hyalonema. There are present a gestral cone, and the body and the root spicule are present here. This is diagram of Hyalonema. And the last class is Demospongia. Demospongia is largest class and more than 90% of the total sponge species. 90% of the total sponge species are included under class Demospongia. They are all marine, but except the freshwater family Spongillida. They have a family which is called as the Spongillida. They are freshwater. Then maximum representative of this phylum are marine water. Only in family Spongillida, which is included under class Demospongia, are freshwater. And the skeleton consists of 1 or 4 raised siliceous spicule. There are also present the siliceous spicule and the spongin fiber or both. Then their skeletal fiber may be made up of spongin fiber or siliceous spicule or both. And the choanocytes are also small. The many important representative of this phylum are Spongilla. Spongilla are also called as the freshwater sponge, and Euspongia called as the bath sponge, Calina called as mermaid's gobes, and the Poterion called as Neptune's cup. So this is the Poterion Neptune's cup, and this is Euspongia and Spongilla, and the Spongilla is the freshwater sponge. Okay, but Cycon is marine water sponge, but the Spongilla is freshwater sponge. In that the freshwater species are present under only class Demospongia. Then, and hope you have understand about the phylum character, the phylum Porifera and their classification. And in the next class, we will discuss about phylum Nidaria from my handwritten notes. So thank you, and that's it.